Okay, we are now on page 14 of the Unit 6 packet. So we're on page Unit 6, page 14. We are going to be talking about why there are problems on the 5' end of the lagging strand. So let's say that we have our DNA. This is our double-stranded DNA. This is the helicase. So let's say that here we have the 3' end right there. That means this is the 5' end. This is the 5' end. This is the 3' end. And then when the helicase moves down the DNA, opens it up, and then DNA replication occurs, DNA is going to be added, and the new DNA strand will be anti-parallel. So let's say that this right here is going to be our new DNA. The new DNA will be added in a 5-3 manner. So this will be the 5' end, this will be the 3' end, this will be the 5' end, this will be the 3' end. There you go. So the green is for this one, this maroon is for this pink one, and in the end it will look like this, but I will show you how we get there. So here we have our strands. They open up, let's say they open up right there. They're going to open up the strands. There we go. DNA is added 5' to 3', meaning that it will go continuously on the side from 5 to 3. And this right here will be the leading strand, which is continuous. And over here will be the lagging strand. So as the helicase moves down the DNA and this opens up, it can only replicate a little bit at a time. So we start from the 5' side to the 3' side. The DNA opens up a little bit more. We go from the 5' side to the 3' side. The DNA opens up a little bit more. We go from the 5' side to the 3' side. The DNA opens up a little bit more. We go from the 5' side to the 3' side. But there comes a point where you can't get to the very end of this side. It's very difficult. So there's going to be a gap there. So what ends up happening is that it looks like this. There we go. So this right here is going to become one DNA molecule, and everything down here becomes another DNA molecule. And there is a gap right here. This gap where there's no DNA. No DNA can get there. And because of that gap, every single time that there is DNA replication, we are getting shorter DNA. And that's really bad for us, because the DNA will get too short over time, and that will lead to the cells not having enough DNA to function, and then the cells die, and this inevitably becomes the reason why we die from old age. So let's say that we look at our chromosome. This is our chromosome with our squiggly DNA coiled up. At the very ends of the chromosome are going to be telomeres. The telomeres are like the aglets on the ends of your shoelaces. So like here is a shoelaces. The ends of the shoelaces are the aglets, and it keeps the shoelaces from fraying. It protects the shoelaces. So let's say that I took some scissors and I cut the plastic part at the end of the shoelace. Well, I can only cut so much until I go into the actual shoelace itself, and then the shoelace starts getting damaged. But there is a little bit that I can cut until the shoelace gets damaged. And so that's the point of the aglet. It protects the shoelace. The telomeres protect the DNA. So the part of the DNA that doesn't get replicated, that's basically the telomeres. And there comes a point where it'll get shorter and shorter and shorter until it hits the DNA that is used for important things for the cell. And right now our telomeres are getting cut down shorter and shorter, so it won't be for another many decades that our regular DNA will be cut down, and then that's when our cells will die, and then we eventually may die from it. And scientists are trying to figure out how to stop the telomeres from being cut. They're trying to figure out how to solve this problem, and they believe that if they solve the problem, then we could live forever. Okay, now on to the second part of page 14, proofreading and repair. So, let's say that mismatch happens. Let's say that we have DNA that is, I don't know, A, T, G, C, C, G, and then that's the parental strand, and then the new DNA strand that we're making is T, A, G, G, C. And so this right here, this is a problem. So let's say that there's a mismatch. Then what would happen is that nuclease would cut it out. So let's say that this paperclip is the nuclease. The nuclease will cut out the A and make it gone. Okay, it goes bye-bye. Takes the A away. And then the DNA polymerase will come in and attach a C there. And then the ligase will glue everything together to make sure it's continuous. So I'll write that down. The nuclease cuts the A out. The DNA polymerase adds the C, and then the ligase glues the nucleotides together. Okay, now page 15. Please attempt the page, pause it, attempt it, and then unpause it in order to get the answers. Hopefully that you did pause it. Now I will go over the answers for you guys. So, okay. Question number 1. DNA replicates during which phase of the cell cycle? That's going to be S phase of interphase. Why is it important that DNA replicates? Because we need a copy of DNA for each of the 2 daughter cells after after cell division. 3, if a parental strand reads that, what would a newly synthesized strand read? So the parental strand is ACGTAC. So the correct way to write this would be, because DNA is anti-parallel, you have a 5 on that end, then you're going to start out with a 3. Boom, we got a 5 on the other end. And then this would be TGCATG. Number 4, what direction does DNA polymerase 3 move along the parental strand? So this is a trick question, kind of, because DNA polymerase will add nucleotides. When it adds nucleotides, the nucleotides themselves are added 5 to 3, but moves on the parental strand 3 to 5. As you can see here, it goes from here to here. So the parental strand is actually 5 to 3. And then question number 5, why do Okazaki fragments occur on the lagging strand? Because the new DNA cannot be added until helicase unwinds more DNA. There we go. That's the last question of the page. Going on to page number 16, that's more of like a review page. I've discussed what all of these things do on a previous page already. Now we're moving on to the next page, page number 17, which is transcription and RNA processing, which is it's a lot going on. So that's going to be for another video.